Class 9 students, welcome to Diksha Vedantu. Now I know your PT tests are here and this is your Aditi Ma'am. I have gotten the most important video that you need for your science PT test. Okay, so make sure you watch the video till the end. Now beta, in this video I am going to tell you some of the most important diagrams of biology of both the chapters, the chapter cell as well as the tissue. These diagrams are need not to be missed at all. Mark my words, all these diagrams are important. Some of them I have given you just as a precaution that if you do that, nothing will be left. But other than these diagrams, you have prepared, right? You have done the whole chapter, the whole question, but these diagrams are very important. Now the first and the foremost beta, we all know your animal cell and plant cell. The basic difference is plant cell has a cell wall, animal cell does not have a cell wall, and based on that you practice the animal cell. See, this is your proper animal cell. We have a nucleus, we have endoplasmic reticulum, your mitochondria, your vacuole. See how clearly this is labeled, right? And then we have our plasma membrane as well. Now on endoplasmic reticulum, you can also label, if you want, not necessary, but you can also label the rough endoplasmic reticulum and the smooth endoplasmic reticulum as well. Okay, and we have so many things. This jelly like substance is your cytoplasm, this is your Golgi apparatus, these small things, right? These are your peroxisome. So all these things are important. Make sure that you study them properly. Okay, next beta, moving on to plant cell. So this video will be very crisp, very clear. We'll just tell you about all the diagrams that you need not to miss. Okay, then moving on to cell wall, we know vacuole is huge in plant cell. We have a different cell wall in plant cell that is absent in animal cell. Inside that cell wall we have our plasma membrane, and then all the things mostly same. We have nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, ribosomes, and our mitochondria, and all the things that are present. We have also have chloroplast in plant cell. Okay, so these differences should be very clear to you. Okay, make sure to do the difference between animal cell and plant cell and draw the diagrams as well. Okay, this is the first thing you have to practice and you cannot miss this. Done. Next beta, the next thing we have hypertonic, hypotonic, isotonic solutions. Why I told you this is important? Because this whole concept is very important beta. We know hypertonic means the cell shrinks, right? The cell shrinks, so water goes out of the cell. In hypotonic it swells up, so water goes inside the cell. In isotonic nothing happens, so the concentration of water remains same. Whatever the water goes out, it comes back. So this is also very important. Okay, next beta, we have plasmolysis. Plasmolysis is if we place the cell in a hypertonic solution, it will shrink, but if we place plant cell in hypertonic solution, it will shrink. But we know cell wall is rigid, so it says I won't shrink, just leave me, I won't shrink. So cell wall remains intact, but the inside of it all shrinks, right? Protoplasm shrinks. Done. That's your plasmolysis diagram is important. Then beta, we have plasma membrane. Now this diagram can look a little bit tricky and hard, but all you have to do is to make certain things very clear. These are your proteins, these are your lipids. That's it. It's a bilayer of lipids and proteins. That's it. Okay, you can make that. This probably won't even come, but just to show you, this is how plasma membrane looks like. And now all the cell organelle is all the nucleus inside the cell. So basically this is the structure. Okay, you can just simply make it like this. Okay, and inside we can clearly see a nucleus. This is the whole nucleus. It is double membraned. This nucleus beta is double membraned, like it has pores on the surface. These are called nuclear pore. Then we have this nuclear envelope around it. The center part is called nucleolus, and we have nucleoplasm just like we had cytoplasm. Done. Then beta, moving on to next, we have endoplasmic reticulum. These are some folds, right? And this is your, this, see, there are some small dots on it. That is your rough endoplasmic reticulum. These dots, these are ribosomes. These, the part where they don't have the small dots, the circles, those are smooth endoplasmic reticulum. Then beta, we have Golgi apparatus, and again sac-like structures, right? Sac-like structures. These are called Golgi apparatus. Then we have lysosome. These are called suicidal bags of the cell. The diagram looks something like this, but you should know the function of it. And if they ask, if you don't know so much lines, you can make this at least, right? These are called digestive bags. Okay, so inside you can just make small enzymes and it will be perfect. Okay, these enzymes come out and the cell dies because of these. Okay, so these are something that is helpful if you don't know the theory. Okay, then beta, we have vacuoles, simply a bubble, nothing, literally. Then we have mitochondria. Now this diagram is very important. First, it is a double membrane, right? It is a double membrane. We have outer membrane. Now inner membrane has folds. Inner membrane has so many folds. It is called intermembrane. The space is called intermembrane space, right? It also has ribosomes. It has its own nucleus. So all these things are very important. Done. Next beta, we have chloroplast. Again, a very important diagram. Coin-like structures. Coin-like structures you need to represent inside the chloroplast. Okay, it again has two membranes. This is the intermembrane space, inner membrane, outer membrane. Now they have stroma and they have their own DNA. They have ribosomes as well, and they have all these structures, but basically they have a coin-like structures inside them. That is the important part. Okay. Next beta, we have meristematic tissue. Very important diagram of your biology. We know it is divided into mainly three parts, three ministers, we remember, right? Apical meristem, intercalary meristem, lateral meristem. Apical meristem is at the roots and the shoots, right? And intercalary meristem is in between the nodes, at the nodes, in between the branches or the leaves. And lateral meristem is increases the girth, the width of the plant. Clear? That is very important. Then beta, we have parenchyma. It has very thin space. Collenchyma, it has thickened corners, right? It has cell wall, it has vacuoles. It is clearly mentioned. Everything is clearly mentioned in all these diagrams. So just go through these, and these are the important ones. Try making them. Okay? And if you see in sclerenchyma, they have very thick walls, right? So these are very important things, right beta? Okay, next we have xylem and phloem. Again, very important. We have tracheas, we have vessel, we have xylem, parenchyma. All these three diagrams are important, and in phloem as well you need to clearly mention these are sieve plates, these are sieve tubes, phloem, parenchyma, these are companion cells. Again, very important. Okay, next beta, we have muscular tissue. In muscular tissue we have two of these, which is your skeletal tissue, also called as striated muscle, right? Then we have smooth muscle. These are straight. Okay, these are straight. Something looks straight. We have nucleuses, so many nucleuses. In here beta, it is a little different, a little elongated in shape, right? Then beta, moving on, we have structure of neuron. Very important. Ma'am, you are saying important to everyone. No, I'm not saying important to everyone, but these things are very important. You cannot miss this. Okay, neuron is, this will definitely come. You mark my words, neuron is definitely coming. It has three parts: the head, the body, and the tail. On the head beta, we have nucleus, we have branches, dendrite, which collect the signal. This is your body. Okay, then we have this axon that carries the signal, and these are the protections of axon, right? Then we have nerve ending through which the signal passes on to the next neuron and or reaches to the brain. Done? Very important. Then beta, moving on to chemistry, we have some very important diagrams. Those are what? The diagrams of the chapter exploration in this chapter, not exploration, in the first chapter, that is your mixtures. The separation part is very important. First is your crystallization. You heat it, you filter it, and you let it stay for a while. The crystals are made. Yes, all these are important. Okay, you can simplify them. You don't have to go into into so much details, but the basic labeling is very necessary. Okay, then beta we have distillation. Now we vaporize the substance, it goes into the air. But what we do in distillation, we trap those vapors, we condenses them in a separate conical flask. Again, not so much detail, but the basic diagram is necessary. Okay, it is necessary. Then fractional distillation, simply make a random box and do this. Okay, do this at each level. These levels you should remember. First is lubricating oil, then diesel, then aviation fuel, then petrol, then petroleum gas. Very important. Next beta we have your paper chromatography. Very easy. Just make a strip of paper, make a dot, and then show that it is kept inside the water, right? That's it. Then beta we have the same paper chromatography but used in the leaves. Okay, now that is something you can easily make. Okay, then beta we have separation of two immiscible liquids. We know oil and water are not mixed together. They cannot, they form a homogeneous mixture. They are separated, right? So this is the technique we use to filter them out. We get the water first in a flask, we take it out, we get the water, and again in a we get the oil again in a different flask. Okay, then beta we have sublimation. Again, naphthalene converts directly from solid to gas, right? So for that we sublime it. The vapors, it gets, the naphthalene gets coated inside the walls of this beaker, and then we take that out. Okay, next beta we have centrifugation. Centrifugation is mainly for colloidal particles like blood. Now if you rotate this blood in a test tube at a very high speed, due to the centrifugal force, all the heavy particles will come at the bottom of the tube and all the watery substance will be there in the whole test tube. So we have separated the thickened blood from the water, the plasma, you can say, right? So that is your centrifugation, and that beta was the all the complete diagrams that you require for your PT test. Now that was biology and chemistry. In physics beta, mostly you will just need graphs. There is a separate video uploaded on the channel for graphs as well. Go watch that video and this video, and you are all sorted for graphs as well as the diagrams. Okay, so beta, make sure not to forget any of the diagrams. This is the video. Make sure you have seen all the live sessions and all the one shots that are there on the channel, and they will definitely help you in your PT1s. Okay, this was your Aditi ma'am. I'll meet you in the next video. This is it.